The CD20-CD3 antibody is a bispecific antibody which has two binding sites for CD20 and one binding site for CD3. This confers increased potency compared to other bispecific antibodies that are currently being investigated, but it gives another particular advantage, which is that it can be co-dosed with other CD20 antibodies, which gives it a pathway for future combinations. So far, we've done a phase 1 clinical trial, and I've reported the updates up until May of the single agent. Because we can co-dose the CD20-CD3 bispecific agent, which we're calling CD20-TCB, with antibody, we're using Gazyva or obinutuzumab as a way to ameliorate cytokine release syndrome and neurological toxicities that are common to this class of agent. What we're doing is giving a flat dose of Gazyva a week before the experimental agent, and then giving the experimental agent every 2 to 3 weeks for between 8 and 12 cycles of therapy. And we did a phase 1 clinical trial. It's actually still underway, still recruiting patients, for patients with relapsed B-cell Non-Hodgkin lymphoma. And it turns out that we recruited a very aggressive population of histologies. 91% were aggressive histologies, around 50% pure diffuse large B-cell lymphoma, and the remainder transformed disease, Richter syndrome, mantle cell lymphoma. And firstly we saw that this seemed to be quite a deliverable agent. We saw that while most patients had side effects, cytokine release syndrome was quite uncommon. It was grade 1 or grade 2 in severity, tended to occur after the first dose of therapy, and then to be much less common with subsequent doses of therapy. There was a relatively high rate of serious adverse event, but this was mostly triggered by patients being observed an extra night in hospital above what was protocolized, which triggered the SAE event, rather than the seriousness of what had occurred. We didn't see very much neurological toxicity at all, only grade 1 or grade 2 and symptoms like headaches, no seizures such as have been seen on other bispecific agents. We saw occasional episodes of tumour flare, and we saw a little bit of neutropenia as well. The neutropenia tended to come on after the first dose of treatment or second dose of treatment, and was more common in patients who'd had multiple prior lines of therapy. As I've mentioned, this was an aggressive group of histologies. Patients that had a median of 3 prior lines of therapy. Remarkably, we saw approximately a 60% overall response rate at the therapeutic doses, and nearly a 40% complete remission rate. We looked at patients who'd had a complete remission to see how durable they were, and at the moment we only have a median of 6 months follow-up, but 71% remain free from relapse or progression. Many of those patients have stopped treatment per protocol but remain in an ongoing remission. So this was the key data that we presented here at Lugano, and I think it's tantalising initial data. The phase 1 study continues to recruit patients. It actually includes an arm which allows Gazyva co-dosing, and we hope to report that data soon. Additionally, there's a trial that's looking at the safety of combinations with R-CHOP, beginning with patients with relapsed disease, but hopefully moving into the frontline setting. And there's a combination trial with atezolizumab.